All right, we are going to finish off our examination of the various antimicrobial drugs that are used. Now that we've looked through the antibacterial drugs, we'll take a look at some of the eukaryotic cells. And this is going to be where the drugs are going to have some problems with selective toxicity because we are composed of eukaryotic cells, and so a lot of the targets are going to be similar, and that's where understanding these fundamental differences in eukaryotic cell structure is going to allow the development of drugs that can be selectively toxic for the pathogen that won't result in those problems with our therapeutic index that's out there. So we'll start out taking a look at our antifungal drugs, and if you remember, there are a couple of different targets that we can look at for fungi that are out there. They do have cell walls. They're different from peptidoglycan. They also have some differences in terms of the steroids that are incorporated in their membranes there. And so if we look at a simple one, we can see this diagram of our cholesterol molecule here. This is the sterol that we're going to be inserting into our membranes to help provide some more hydrophobicity to it and stabilize that membrane in an aqueous environment. And when you look at your fungus that are out there, they're going to be inserting this ergosterol. So it is still in this steroid category, but it is different from the human one. And so that difference is going to give you that selective target to try and interfere with the integrity of your membrane there for your cells. And when we look, there are a couple of drugs that are going to be involved in this. One is this polyene drug over here, and these are trademarked as nystatin or something like amphotericin B. And what they will do is kind of interesting. They will actually insert into the membrane and sort of line up along the side of that ergosterol that you find in the membrane of the fungus cell. And when they line up there, they will actually end up poking a hole in the membrane. And so here you disrupt the membrane, and that's going to result in the cells leaking their cytoplasm, and that can be toxic to the cells. So there are some slight problems with this because of that similarity to cholesterol. So you do have to be a little bit careful with the application of these, and oftentimes they are going to be used topically. When you look at the other side of this, we actually do have a variety of different drugs that are out there that can be used to disrupt the synthesis of the sterol molecule in the fungus cells. And so again, if you don't have that steroid there, you're going to be disrupting the integrity of your membrane there. If we look at the cell wall, again, there are some targets that we can find there. Your echinocandins over here are actually going to interfere with the synthesis of the sugar molecules that are going to be used to make the cell wall for your fungus cell right there. And if you have that, it's going to be similar to what we see with our peptidoglycan cell wall disruptors. In the absence of that cell wall, the cell is going to be much more sensitive to osmotic lysis, and as opposed to just leaking out the cytoplasm, now the cells are going to be lysing out there. There also can be chitin in the cell wall for fungus cells, and so we have another target out there for these polyoxins or nicomycins that are out there, can interfere with your cell wall synthesis. An interesting one over here, griseofulvin, is going to actually interfere with microtubule formation. And if you think about that, if fungus does not have microtubules, what's going to be moving around the chromosomes during mitosis and cell division there? So that's going to result in really the death of your cells. So this will be fungostatic or fungicidal if you're killing off the cells that are out there. You do have to be a little bit more careful with this drug because it does have some toxic effects on your liver, so it's going to be hepatotoxic if it reaches the right dose. But also remember, your liver is going to be filtering some of these drugs out of your bloodstream, so it can accumulate in much higher concentrations. So it might be at the minimal inhibitory or fungistatic concentration, but if it's concentrated, then you can get some damage to the other tissues in the body there. The last one that we can really pull out of here is an interesting one because this relies on a biosynthetic pathway that is different in the fungus cell compared to our cells that are out there. And this fluorocytosine molecule is actually going to be converted by an enzyme inside the fungus into a chemical that's toxic. So here, the administration of the drug isn't going to cause problems to us, but in a fungus that absorbs the drug, it's actually going to take what's known as the prodrug and convert that into the toxic chemical there. So really kind of an interesting thing, because that'll interfere with DNA replication and ultimately protein synthesis inside of the cells there. If we switch gears and start to take a look at the table here, you can see a nice little summary for those, and we see a similar kind of summary when we look at our anti-protozoan drugs that are out there. And this is where we really start to run into some problems with specificity, because your protozoan cells are going to be a lot more similar compared to the other microbes that are out there to our cells. And so you can see just a wide variety of different kinds of specific mechanisms of action, from disrupting your electron transport in mitochondria. And if you think about that, that's going to result in a massive loss of energy, because if you remember from our metabolism chapter, for every glucose that goes in, you have the potential of making 36 ATP in a eukaryotic cell. 34 of those are going to be produced in the mitochondria through that electron transport chain and chemiosmosis. So the administration of this is really going to block the energy production inside of cells. We also have our folic acid synthesis that we saw before with our sulfa drugs that are out there. And artemisinin is really kind of an interesting one that's used often as a cocktail for treating malaria, because this one actually results in the production of what are known as reactive oxygen species that are going to cause oxidative damage to the protozoan, and this is often used with malaria to get rid of plasmodium that are out there. When we look at some of these guys down here, again, we can see different effects from inhibition of DNA synthesis here. And when you look at these drugs, they actually tend to work in microaerophilic environments. And it turns out in low oxygen environments, these guys will actually result in breaks in the DNA. And so that can disrupt the cells and cause death of the cells that are out there. So as long as there's plenty of oxygen around, you shouldn't have to worry. But this is going to work for our microaerophilic kinds of organisms that are out there, like your Giardia or your T-vag, that are growing in these low oxygen environments that are out there. And then the last one is something that we have recently been exposed to in the media, because when you look at these quinolines like chloroquine, this is something that is an interesting drug that nobody is really sure about the exact mechanism, but they do know it's going to interfere with heme groups that you're going to find in red blood cells. And the side effect is it actually is going to be toxic to a variety of different protozoan cells that are out there. When we look at our worms, again, we're working our way up closer and closer, because now we've actually entered the animal kingdom. And these are going to be very similar to a lot of the mechanisms that you'll find in our cells. And oftentimes these are going to rely on the ability of your intestines to not allow the drug to be absorbed and enter your bloodstream, whereas a lot of the worms that are hanging out in your intestines are absorbing their food through their skin, and so not only will they absorb the food, but they will actually absorb the drug at the same time. And you can see we've got a variety of different things, from microtubule formation being inhibited to reducing glucose uptake, and that is going to basically result in paralysis of the worm, and also a lack of energy because they can no longer get their glucose in there. With the avermectins, you can see, again, paralysis going on there for those cells. And then a variety of other things that are going to target things from ATP synthesis to calcium influx that's going to be necessary for these guys to actually have the proper electrolyte balance. All right, the very last thing that we have in this chapter for our drugs that are out there is the antivirals. And this is really, really difficult to develop drugs that are going to have selective activity or selective toxicity against animal viruses, because our animal viruses are hijacking the host systems. And so being able to identify a target that is really selective is going to be a lot more difficult, because we need to find something about the virus replication cycle that is actually different from our cells. So oftentimes you will look for specific kinds of enzyme steps that are there. And if you think back to the viral infection and replication cycle, you can look at they need to adhere to cells, so you've got a target right there in terms of your spikes attaching to the host cell receptors. They need to penetrate and get inside of the cell, so there's another target. And then you can try and find targets in terms of the biosynthetic portion or the assembly phase. So a lot of different things that you have to find the specific sites in the viral replication where you can actually interfere. And you can see a variety of different examples here on this table where we can see the antiviral drugs with their selective toxicity. And I want to show you one specific example of this with our nucleoside analog that is out there with Acyclovir, that is actually very, very specific for herpes virus infections. And if we look at our nucleoside analog for Acyclovir, here is your guanosine nucleoside, and notice this does not have any phosphates added to it on the 5' carbon right there. But we do have our guanine base, we've got our 5-carbon sugar, and then we've got this 5' carbon where the phosphates can be added. And this is what actually makes it selective. And then on the opposite side of the guanosine over here is where we run into the toxicity part. And if you look at this guanosine compared to our Acyclovir molecule, notice we still have the G base, but our sugar is a little bit messed up. It's missing the bottom half of the sugar right there. So you still have the equivalent of your 5' carbon where you can add your phosphates, but the thing that is missing is the 3' carbon where you actually have your hydroxyl group for the next base to add. And if we look at how Acyclovir is actually able to block herpes virus, we can see in this example right here that it's selective because there's actually a viral enzyme that is going to add phosphates to that 5' carbon of the Acyclovir. And so if we look at this pathway, there's your Acyclovir. The viral kinase enzyme adds a phosphate, and then human enzymes are going to add the second and the third phosphate. And this thing, remember, it has that G nucleotide base on it, but now we've got our 3 phosphates and half of that sugar. So now you've got an active drug, and it's only going to really be found in high concentration in cells that are infected with the virus, where they're actually producing that viral kinase enzyme right there. Our human enzymes that would normally catalyze this reaction tend to ignore the Acyclovir because they don't have that full sugar right there. So it's almost like our enzyme not only fills the G base, but we also are going to feel around to make sure that the full sugar is right there. So now that you've activated this drug, you've got your selectivity built into it. We want to take a look at, well, how does that make it toxic to the cells? And if we look at our toxicity, that's the other side of this figure right here. And as the viral DNA is being replicated, and herpes viruses that are out there, things like cold sores and chickenpox types of viruses, those guys actually have not only their own viral kinase enzyme to add phosphate, they actually have their own DNA polymerase that they will use to copy their DNA. And as that viral DNA is being generated there, when the Acyclovir is inserted into the growing DNA strand, it's going to result in what's known as chain termination. The Acyclovir is lacking that 3' hydroxyl group, and that means that the polymerase has nowhere to add the next base onto its growing DNA strand right there. And so that will terminate the chain as it's being synthesized, and without your full-length virus DNA, that's going to disrupt the whole viral replication cycle right there. And so this is where we get selectivity, because the enzymes recognize the drug and make it the active form, and the toxicity because of the chain termination. And the human kinase and polymerase tend not to use these tend not to produce the activated form of acyclovir, and also tend not to use it in DNA replication. There are some side effects. You get some nephrotoxicity, damage to your kidneys, and also this can be slightly mutagenic. But again, it's one of those things, is the damage that might be produced as a result of the drug worth getting rid of the infection that can cause even more damage that's out there? And if we look at something like retroviruses that are out there with something like HIV, you can see this is actually a drug cocktail that comes into play to block different steps in the replication of the virus here. And it's known as highly active anti-retroviral therapy, or HAART. And you can see a variety of different targets that are out there, from targets that'll block fusion of the virus with the receptor on the cells, to the reverse transcriptase inhibitors over here like AZT, and you block the virus from being able to change the RNA into DNA. There is inhibitors that block the integration step, and then inhibitors that block what are known as protease activity, where the long protein that is made by the cell with this HIV protein, the protease inhibitors will prevent it from being chopped up into little pieces, and without the pieces you're not going to have all of the proteins that are necessary to make your fully functional HIV virion that can bud off of the cell. So you might be able to get rid of HIV by using any one of these drugs by itself, but by using this cocktail, you increase the chances of getting rid of the virus by blocking it at multiple different steps. And it's less likely to develop resistance to all of these different steps at once. But viruses are known because they do have polymerases that are error prone compared to our cells. So when the virus is making hundreds of progeny virions, it's not really a problem for the virus if a few of them are messed up, because a few of them might actually develop mutations that allow them to resist the activity of these different kinds of drugs. And that's where we want to take a look at this idea of the development of these drug-resistant kinds of microbes. And if you look at this figure, you can see we've got a population of cells that are out there. And normally you could have, billions of these cells floating around, and some of them might actually be resistant. They have developed a mutation that allows them to resist the drug. But those aren't really going to become the prominent members of the population until you put selective pressure on those drug-resistant mutants, and then you kill off all the sensitive cells, and then the ones that are going to be left over are the ones that are drug-resistant. And so now you've got a whole bunch of those cells making up that population. And think about the stuff that we learned in chapter 11 about all the different ways that DNA can be shuttled from one cell to another, whether it's by transformation, transduction through a virus, or conjugation, where the donor is going to give that chunk of DNA to a sensitive cell that's out there, and then both of them are going to be resistant as a result of that transfer of the genes through that horizontal transfer as opposed to vertical transfer. And when we look at this, you can think about, well, what can contribute to that selective pressure? Well, one of them is natural selection. These drugs are providing selective pressure to select for the resistant cells. We've also got this overuse and misuse of antimicrobials, like we saw in the earlier graph I showed, where people are asking for antibacterial drugs to treat influenza, when all they're going to be doing is getting rid of the bacteria in their intestines, because they're taking a broad spectrum to try and treat a viral infection. We've also got subtherapeutic dosing in things like animal feed, where people are administering these drugs to the animals that are out there, and then the population of cells develop resistance, and then we can get exposed to those later on. But the other major thing to worry about is people. You have people, when they're given a prescription, and after a few days they feel better, and so they stop taking the drug, and then they keep it for months, and then when they get sick again they go ahead and take that drug. Well, maybe it was at the minimal inhibitory concentration when they first had it, but after a few months or even a few years the potency of the drug might have been reduced, so now you're using a sub-therapeutic dose, and it's no longer going to work to get rid of that infection. Or maybe you give it to your friend because they're feeling sick, and you want to help them out. And that's really one of the things that is resulting in more and more drug-resistant kinds of cells developing, and then that is going to really cause problems for treating these kinds of pathogens in the future. The final thing we'll look at is, well, what kind of mechanisms can be used to develop these drug resistance within the cells that are out there. And these are some really novel kind of evolutionary advancements that the cells are coming up with, from things like efflux pumps over here, which are actually just a protein that's inserted in the membrane of the cell, and its job is actually just to sit there and pump these drugs out of the cells. And as long as that drug isn't present there, you don't have to worry about it causing death or inhibiting the cell. And when you look at something like tetracyclines, they actually have an operon. As long as the tetracycline is around, it'll bind to a repressor, kind of like what we see with lactose, and that repressor falls off the DNA and it turns on the gene to make your tetracycline pump, and then the cells are going to pump that out. And as long as the tetracycline is there, they're going to continue making this efflux pump. We can also see things like proteins or genes inside of the cells that will actually code for enzymes that will modify the drug. And if your drug can't bind to your target, or if you break down the drug, like your beta-lactamases that are out there, those enzymes are actually going to break the beta-lactam ring, and if you don't have the beta-lactam ring, it can no longer bind to those proteins that are involved in the transpeptidation reaction to form peptidoglycan. We can also see a couple of other interesting mechanisms for developing drug resistance. You can even modify the target inside of the cells. So what if you change your penicillin binding protein so that your penicillin no longer binds there, or change the structure of your ribosome so that the drug can't bind there? Another common thing that we can find out there is what's known as overproduction, where you develop a mutation that results in the promoter being more effective, and that means that you produce more of the enzyme, and so maybe you can do this sort of mass action where you produce so much enzyme the drug can no longer block it out there. Or maybe you develop a mutation to allow an alternative pathway that would allow you to get around the reaction that's been blocked. And a really interesting one that's out there that people have found is what's known as target mimicry. And here you actually get the targets that are normally bound by the drugs are going to be mimicked by something else. So imagine if you make a whole bunch of extra protein and your drug binds to that thing. So almost like a decoy to prevent it from binding to the actual target. And we can find this with tuberculosis. They actually make a protein that mimics the structure of the DNA, and your fluoroquinolone drugs will go in there and bind to this thing instead of binding to your DNA gyrase that is used to relax the supercoils for DNA replication to occur. You can also find with the aminoglycosides that are out there that those drugs will block the A site in the large subunit of your ribosomes. Well, cells can actually make a protein that mimics the shape of that A site in the ribosome, and now your aminoglycosides are going to preferentially bind to this target that is kind of the same shape, so that your drugs are doing what they're supposed to. They're just binding to the wrong thing. And just to sort of show you, these drug-resistant microbes are really becoming a bigger and bigger problem. And the CDC estimates that we're looking at almost 3 million infections of antibiotic-resistant microbes every year, and this is resulting in more than 35,000 deaths. And this can range from things like MRSA, where you've got your methicillin-resistant Staph aureus, and these guys are producing these penicillin-binding proteins that are out there that do bind to the drug and make it no longer effective. to things like vancomycin-resistant Enterococcus and Staph aureus that are out there. We've got some gram-negatives that have not only beta-lactamase enzymes, but they can actually make what's known as a broad-spectrum beta-lactamase that inactivates all of the beta-lactam antibiotics that are out there. So there isn't really a good way to treat these. When you look at some other Cre bacteria, these are gram-negatives that are actually pumping the drugs out of the cells there. And so really kind of nasty different kinds of things if we think about the exchange or horizontal transfer of these genes from one cell to the other. And what we're finding now is multi-drug resistant bacteria are actually evolving. And when you look at the bacteria that causes your sexually transmitted disease gonorrhea, that thing has developed resistance to almost everything that is out there. And we really only have one antibiotic left to treat this, and that antibiotic has some serious side effects. And so again, it's that question of efficacy. Is it worth the damage that might occur compared to the damage that might occur from the drug as opposed to the damage that occurs from the pathogen? And when you look at tuberculosis, there's multi-drug resistant bacteria, and now a new one that is called extremely drug-resistant bacteria. And when you look at these guys, not only are they resistant to isoniazid, they're also resistant to rifampin, and they're resistant to fluoroquinolone, and the last second line of drugs that we have out there that are injectable that can cause some damage. So more and more, this is something that we're going to have to worry about in the future as we keep putting selective pressure on these pathogens, and they're developing resistance much quicker. To give you sort of an idea, they discovered penicillin and started using it, and within a year they had already found drug-resistant bacteria out there. So this is definitely something we're going to have to worry about in the future.